Igor Nelson is an independent researcher in energy, biology, and living systems, leading VortexLab, a research initiative dedicated to exploring the connections between energy and living systems. This presentation provides a summary of key ideas related to bioelectricity, electromagnetic fields, Schumann resonance, and their possible influence on human health and biological regulation. The full scientific discussion and references are available in the article. Exploring the influence of Schumann resonance and electromagnetic fields on bioelectricity and human health. Extremely low-frequency electromagnetic fields, or ELF/EMF, are electromagnetic oscillations generally found between 3 and 30 Hz. These frequencies occur naturally in the Earth's environment through lightning activity, geomagnetic fluctuations, and interactions between the Earth's surface and the ionosphere. One of the most relevant natural ELF phenomena is the Schumann resonance, a set of global electromagnetic resonances with a fundamental frequency close to 7.83 Hz and higher modes around 14.3, 20.8, 27.3, and 33.8 Hz. These frequency ranges partially overlap with human electroencephalographic activity, especially theta, alpha, and beta brainwave bands. From an evolutionary perspective, biological systems have been continuously exposed to natural electromagnetic fields. This long-term exposure may have contributed to adaptive mechanisms that allow organisms to respond to environmental electromagnetic variations. The text suggests that ELF/EMF may participate in the regulation of physiological processes such as circadian and circaseptan rhythms, blood pressure, heart rate variability, and melatonin secretion. Melatonin is particularly relevant because it is involved in sleep regulation, endocrine function, oxidative balance, and broader biological synchronization. The central scientific question is how ELF/EMF affects biological systems and which mechanisms mediate these effects. Proposed mechanisms include interactions with cellular membranes, modulation of voltage-gated calcium channels, changes in intracellular calcium concentration, alterations in resting membrane potential, and effects on intracellular signaling pathways. These mechanisms are important because bioelectric activity is involved not only in nervous system function, but also in cell communication, tissue repair, morphogenesis, and organismal development. The topic has become increasingly relevant in modern life because natural electromagnetic fields now coexist with artificial sources of electromagnetic pollution, including power lines, domestic appliances, industrial equipment, and technological infrastructure. These artificial fields may overlap with or interfere with natural ELF signals. At the same time, variations in geomagnetic and solar activity have been associated in several studies with changes in cardiovascular function, melatonin levels, neurological activity, and other physiological parameters. Research reviewed in the text reports several biological effects associated with ELF exposure. These include modifications in brain electrical activity, changes in circadian rhythm regulation, reduced melatonin production, increased intracellular calcium, wound healing effects, and possible associations with cancer-related processes. Some studies also suggest that specific ELF frequencies, including frequencies close to the Schumann fundamental mode, may produce beneficial cellular effects, such as inhibition of certain cancer cell models in vitro. However, these findings remain complex and context-dependent, since biological outcomes may vary according to frequency, intensity, exposure duration, and the physiological state of the organism. A further area of interest is space physiology. During missions to the Moon or Mars, humans may be exposed to electromagnetic environments that differ significantly from those on Earth. Since natural Earth-based ELF signals may contribute to biological synchronization, their absence or alteration could have implications for astronaut health, circadian regulation, and psychological well-being. In conclusion, ELF/EMF and Schumann resonance represent a potentially important interface between environmental physics and biology. Current evidence suggests that these fields can influence biological systems at cellular, physiological, and systemic levels. Nevertheless, the underlying mechanisms remain insufficiently understood. Further experimental research is required to distinguish beneficial from harmful effects, clarify dose-response relationships, and determine how natural and artificial ELF exposure affects human health. Bioelectricity is a fundamental property of living organisms. Cells and tissues generate endogenous electromagnetic fields that participate in essential physiological processes, including nervous system signaling, muscle contraction, cardiac activity, sensory perception, cell communication, embryonic development, wound healing, and tissue regeneration. The main source of bioelectricity is the movement of ions across cell membranes. This movement generates the resting membrane potential, or RMP, which depends mainly on ionic concentration gradients and membrane permeability. The most important ions involved are potassium, sodium, calcium, and chloride. Traditionally, changes in membrane potential were mainly associated with excitable cells, such as neurons and muscle cells. However, recent research indicates that RMP is also highly relevant in non-excitable cells, including skin, bone, and other tissues. In these cells, membrane potential contributes to cell volume regulation, ion balance, nutrient transport, and intracellular signaling. A particularly important finding is that healthy cells generally maintain a more polarized membrane potential, whereas dysfunctional or diseased cells tend to be more depolarized. This is especially relevant in cancer biology. Studies by Yang and Brackenberry suggest that highly proliferative cells, including cancer cells, usually present lower membrane potentials, while non-dividing or differentiated cells show higher electrical polarization. This suggests that RMP may serve as a useful clinical marker for tumor detection and prognosis. More importantly, bioelectric states may not only reflect disease, but also influence disease development. Research by Michael Levin and colleagues supports the idea that bioelectric fields can act as control signals in morphology, regeneration, and cancer suppression. In experimental models, correct bioelectric regulation was able to prevent disease expression even when cancer-associated genetic factors were present. Overall, bioelectricity appears to be a key regulatory system in cell function and organism development. Resting membrane potential is not only a consequence of cellular activity, but may also help determine cell behavior, proliferation, differentiation, regeneration, and pathological transformation. Extremely low-frequency electromagnetic fields, or ELF/EMF, can influence cellular function, although the mechanisms remain only partially understood. Proteins are not usually expected to resonate directly at very low frequencies, such as 7.83 Hz, because molecular resonance typically occurs at much higher frequencies. However, ELF/EMF may still affect biological systems indirectly by modulating signal transduction pathways, especially through ion channels. One of the main proposed mechanisms is the activation or modulation of membrane ion channels. Depending on frequency, amplitude, exposure duration, waveform, and tissue-specific properties, ELF exposure may alter the opening probability of these channels. This can affect electrical signaling, resting membrane potential, ion homeostasis, cell volume regulation, intracellular communication, and gene expression. Calcium signaling is particularly important. Voltage-gated calcium channels, or VGCCs, represent one possible pathway through which ELF/EMF may affect cells. When calcium enters the cell, it acts as a second messenger involved in muscle contraction, neuronal activity, cell division, gene expression, apoptosis, intracellular transport, and membrane dynamics. Several experimental studies report increased intracellular calcium after ELF exposure. For example, exposure to low-frequency electromagnetic fields has been associated with increased calcium uptake in normal and leukemia lymphocytes. Other studies reported that ELF exposure inhibited proliferation in several cancer cell lines and induced changes in cell cycle regulation, organelle morphology, immune signaling, and apoptosis. In some models, these effects were linked to rapid calcium influx and activation of apoptotic pathways. ELF effects have also been observed in stem cells and plants. Pulsed electromagnetic stimulation around 7.5 Hz has been associated with osteogenic differentiation in human mesenchymal stem cells, possibly through calcium channel activation. In plants, Schumann resonance-like frequencies may influence photosynthetic parameters, although the effect appears to depend on the species and may also involve calcium-related mechanisms. However, an important limitation must be emphasized. Natural Schumann resonance magnetic fields are extremely weak, usually in the picotesla range, which is far below the Earth's static magnetic field. At these intensities, there is no fully established classical mechanism explaining direct activation of membrane ion channels. Therefore, alternative mechanisms have been proposed. One of the most plausible current hypotheses is the radical pair mechanism, which may allow weak magnetic fields to influence biochemical reactions involving reactive oxygen species. Recent evidence suggests that mitochondria may be a primary site of ELF interaction. Through changes in mitochondrial redox balance and ROS signaling, ELF exposure could indirectly affect calcium dynamics, membrane potential, and broader bioelectric regulation. Overall, ELF/EMF may influence cellular behavior through ion channels, calcium signaling, mitochondrial activity, oxidative balance, and intracellular signaling pathways. Nevertheless, the precise mechanisms remain uncertain, especially at natural environmental field strengths. Further research is required to distinguish direct electromagnetic effects from indirect biochemical responses and to clarify their relevance for health, disease, and bioelectric regulation. Biological systems may have evolved under continuous exposure to the Earth's natural extremely low-frequency electromagnetic fields. Since the brain, heart, and other organs operate through electrical and electromagnetic oscillations, it is possible that some physiological processes became partially synchronized with natural ELF signals, including Schumann resonance. The central hypothesis is that the fundamental Schumann resonance frequency, around 7.83 Hz, may be especially relevant for biological regulation. This frequency may interact with cellular bioelectricity and potentially influence resting membrane potential, cellular communication, and organism-level synchronization. Traditionally, the biological effects of ELF have been explained mainly through calcium signaling. According to this view, ELF exposure may modulate calcium influx and efflux through ion channels, particularly voltage-gated calcium channels. This could affect resting membrane potential and influence several downstream processes, including cell signaling, apoptosis, differentiation, and tissue regulation. However, recent evidence suggests that calcium signaling may not be the primary mechanism. A more updated hypothesis proposes that weak ELF fields may first act through the radical pair mechanism, especially within mitochondria. Mitochondria are central to reactive oxygen species production and redox regulation, and changes in mitochondrial ROS dynamics may indirectly influence calcium signaling, membrane potential, and broader bioelectric states. This model is important because mitochondria are physically and functionally connected to calcium regulation through mitochondria-associated membranes. Therefore, ELF-induced changes in mitochondrial redox balance could act as an initial trigger, while calcium changes may occur as a secondary downstream response. At the neurological level, Schumann resonance may also be related to brainwave entrainment. The thalamic rhythm generator, located in the thalamus, plays an important role in regulating brain oscillations. During silent phases of thalamocortical neurons, low intracellular calcium levels may make these cells more sensitive to environmental electromagnetic fields. If atmospheric ELF frequencies synchronize with thalamic rhythms, this could help explain similarities between Schumann resonance frequencies and some EEG patterns. This entrainment hypothesis suggests that natural ELF signals may contribute to the coordination of brain activity, circadian rhythms, and possibly states of consciousness. In contrast, artificial or unnatural electromagnetic fields could disturb these rhythms and contribute to abnormal EEG activity, disrupted biological cycles, or irregular cellular function. Another important point is that many subcellular structures already display rhythmic activity. Examples include mitochondrial membrane potential oscillations, calcium flashes, and cytosolic redox cycles. These biological oscillators may interact with external ELF fields depending on frequency and phase alignment. This could explain why biological systems may be selectively sensitive to narrow frequency bands, such as Schumann resonance. From a biomedical perspective, bioelectricity may represent a regulatory system that operates alongside genetics. If specific ELF frequencies can influence cellular bioelectric states, controlled electromagnetic exposure could eventually become relevant for cancer treatment, tissue regeneration, wound healing, or modulation of abnormal resting membrane potential. Finally, the discussion has implications for space physiology. During missions to the Moon or Mars, astronauts will be outside the Earth's natural electromagnetic environment. For this reason, simulated Schumann resonance fields may be considered as a possible strategy to support biological synchronization, homeostasis, and long-term health during space travel. Overall, ELF-organism interactions may involve several interconnected mechanisms, including mitochondrial redox signaling, radical pair reactions, calcium dynamics, membrane potential regulation, and brainwave entrainment. Although the evidence is promising, further experimental research is required to confirm these mechanisms and determine their physiological and clinical relevance. This presentation was intended as a brief summary of the main ideas discussed in the full article, highlighting how Schumann resonance, electromagnetic fields, and bioelectricity may interact with biological systems and human health. The key message is that living systems are not only biochemical, but also bioelectrical and informational. Understanding these interactions may open new perspectives for health, regeneration, cellular regulation, and integrative biomedical research.